We've called this DNA and genetic engineering the basics. So hopefully after tonight you guys will have an idea of what's going to be involved in your iGEM project. So starting off, this is a nice cartoon representation of what DNA is. DNA is a double-stranded helix that's made up of base pairs between chemical compounds. And DNA has 4 nucleotides, what we call them, that make up DNA, and they have specific properties about them that allow them to form this helix. So there's adenine, thymine, cytosine, and guanine, and we just abbreviate those to A, T, C, and G. And you guys have probably heard of those bases before, right? Yes? No? Yes? If you not, that's good. Okay, so what those bases are made up of are 3 different components. So first we have the base itself, and that gives it the letter that we've abbreviated them to. They also have a sugar molecule that it's attached to, and a phosphate molecule. And those 3 things are what make up the bases and what it needs to connect them into this double helix. So, so A and T can base pair with each other based on the properties of the base, and C and G can base pair with each other. And that's kind of why DNA is double-stranded. So as long as you have one strand of DNA, the cell knows what the other strand has to be, because only T can pair with A and only G can pair with C. So you only ever need one strand, but it comes as 2, as kind of like a double copy of the same information. So DNA also has a direction, and we call that direction 5' to 3'. And that's based on the sugar molecule that it's attached to. So when we number the atoms in a sugar molecule, we give them, a sequential number. So starting in this corner, it would be carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So this carbon down here is the 3' carbon, and the carbon up here is the 5' carbon. So in the DNA helix, the chain will continue from the 5' end through the 3' end, and this is where the next base is going to attach. So when we say that DNA has a direction, it just means that we're going from the phosphate on the 5' side down to the 3' side of the sugar. And then DNA is also anti-parallel, and what that means is that the 2 strands run in opposite directions. So the top strand will go from 5' to 3' this way, but the bottom strand will go 5' to 3' the other way. And that's based on how those base pairing interactions have to form. Everybody still following so far? No questions? Okay. DNA comes in a couple different forms, especially in bacteria. So bacteria have genomic DNA, and that's what every cell has. The genome is what tells the cell what kind of cell it's going to be, what kind of proteins it's going to make, what it needs to function and be alive. But bacteria can also have what's called plasmid DNA, and that's kind of extra pieces of DNA that bacteria can grab from the environment, that they can pass on when they replicate, and it's just a circular piece of DNA that can encode for extra genes. And plasmid DNA is very fortuitous for us in iGEM. It's what we use all the time to help engineer these cells, because if we can put specific DNA onto a plasmid and give it to the bacteria, then we've engineered them to have that gene that maybe they didn't have to begin with. So before we can start engineering these cells, we need to be able to read DNA. And DNA is really simple to read. It has a universal genetic code, and that means, universal means that every living thing, every cell uses this same code. So if it's a bacteria, if it's a plant or an animal, every animal or every cell that has DNA uses this same code. And it basically works in groups of 3 nucleotides, or 3 bases. So up here, the sequence TTT will code for 1 amino acid. In this case, it will be phenylalanine. That's an amino acid that will be used when the gene is translated into a protein. letter sequence has a deeper meaning on the protein level. So when DNA gets transcribed into RNA, it'll keep these three-letter codes, and then the cell will use that code to make the protein and string a whole bunch of amino acids together, and that'll give us our protein at the end. So this is a little more about gene expression. So that's basically the formation of a protein. So we're going to start with our DNA that carries the code that we need to make a protein. DNA can be replicated, so it can use the double-stranded nature of DNA to make a double copy of itself. And the cell needs to do that when it replicates, because if it's going to divide in two, it has to make sure it still has all that DNA and all the genetic information that it needs to keep living. So it'll replicate it, make a copy, so that each cell can have one copy of that DNA. DNA can also be transcribed into RNA. RNA is like the sister molecule of DNA. It's very, very similar in structure. Has a few different properties that make it perform different in the cell. And RNA will still have those same base pairing rules as DNA. But RNA can also be recognized by the ribosome in the cell. And the ribosome is a very important machine that the cell uses to make proteins. So the message in the RNA is then translated into the protein product. So here's kind of an analogy for you. You have a master copy of, let's say, a document that you've made for school. You can make a photocopy of that, and that's kind of like your RNA, and then the product is the same as the protein. So if you're going to bind the pages together into a book, that would be your protein. Good, no questions yet? Okay, so here's a little bit more about plasmid DNA and why it's so awesome to use. Basically, we use it as a vehicle to deliver different genes and DNA pieces. So we can put a sequence into a plasmid DNA and give it to a bacteria to use to make proteins. One very important thing that plasmid DNA has are genes for antibiotic resistance. And well, that might not seem right at first. You know, if you have a bacteria and you can't treat it with antibiotics because it's resistant, then you can't really kill it, right? Well, it's good in our setting where we can control it. So normal bacteria don't have resistance to antibiotics, and that's why we can take antibiotics like from the doctor as prescriptions and kill bacterial infections. But if we want to engineer our bacterial cells, we want to be able to distinguish between the ones that have a plasmid and the ones that don't. So if we take a plate where we grow bacteria on and add antibiotics to it, normal bacteria won't grow. If we give them a plasmid with a special gene and an antibiotic resistance gene, it now has the information that it needs to survive on that plate, and the antibiotics won't kill it. So we call that a screening method, so that we can tell which bacteria have the plasmids that we gave them and which ones don't. Does that still make sense? Yeah? Okay. This is just a cute comic I found that kind of describes that a little better. So the shady figure in the dark alley, he says, "Hey kid, want to be a superbug? Stick some of this into your genome. Even penicillin won't be able to harm you." And on the bottom it says it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. So basically, bacteria can share pieces of DNA. So if this bacteria has a plasmid with the gene for antibiotic to say, or antibiotic resistance to penicillin in this case, it can transfer that DNA to another cell, and that cell will then also have resistance to penicillin. So that's kind of a cute comment. Maybe it makes a little more sense. Yeah. So when we're engineering a cell, we kind of call it assembling DNA. So how do we assemble DNA? How do we engineer it? Well, first of all, we need to have the gene that we want to give the cells. So we have to make that gene. So there are companies that will actually synthesize it for us because we know what the code is, we know how to read and how to write DNA. We can send the DNA sequence to a company, they know what to do, they put all the base pairs in order, and they make the gene for us and they send it back to us. That's really easy. There's also another way that we can make a gene, and it's through a process called PCR, which is abbreviated, the abbreviation for polymerase chain reaction, and Anthony's going to go over that very soon. So once we have the gene that we want, we can also make the plasmid that we want to put that gene into. And the easiest way to do that is to grow bacteria that have that plasmid, and they'll just replicate it like they do all their other DNA with all their cellular processes. So we grow bacteria, they make the plasmid for us, and then we can isolate the plasmid from the bacteria once they've grown long enough. Then we can basically cut and paste the DNA together to make one final product with our plasmid that has our new gene in it. And then we'll transform that plasmid into bacteria. That basically just means that we're going to give the plasmid to the bacteria so that they have this new piece of DNA. And how we do that is basically just stressing the cells. So if we stress the cells, like heat them up to a little bit higher than their normal temperature, they kind of start freaking out saying, oh no, what am I going to do to survive? It's not the optimal conditions for me. They kind of just start reaching out and grabbing whatever kind of DNA they can. Hopefully that DNA that they grab has some kind of gene that will help them survive. So this is a very simple technique we use to help give the plasmid to bacteria. We heat them up a little bit, they freak out, they start grabbing what kind of DNA they can get, and in that way we kind of get that plasmid DNA into the bacteria, and then they can start using the new genes that we gave them.
Okay, so Jenna mentioned PCR, and that is a polymerase chain reaction. So this kind of looks a little overwhelming, but essentially what's happening is you start out with your DNA, which is double-stranded, and if you heat that up, the two strands will actually come apart, okay? And so that's what's shown here. And what we use are what are called primers. And that's essentially a short piece of RNA, which is stuff in the red, and that matches up in sequence with the DNA. And not RNA, DNA. Okay, this is all DNA. Okay, so we have a primer, that's a short sequence that will pair up with the already template DNA. And what happens is, if you remember back at the beginning when we were talking about how cells actually replicate their DNA, like normally, because they have to make another copy for when they split into two. And using that same machinery, you can actually essentially use these primers and extend them, and then you have, from here you have one piece of DNA that becomes two that become four become 8. So each time you heat it up, they'll split it apart, and then use the machine, they'll grow the length of DNA, and then again you heat it up again, and then you keep repeating the process and get a whole bunch of the same DNA. Does that make sense? Okay. So essentially, with iGEM, they have a registry of what they call biobricks or parts, and here are a list of classifications they use for these parts, but all these is essentially DNA sequences. And using the biobrick assembly method, which I'll be talking about very soon, you can actually string them along in any order you want them to, or you want to put them in. So here we have some pictures that they actually use to represent these parts. So we have like this 90-degree arrow, which actually represents a promoter, ribosome binding site. This is just a porter of some sort, and this is a terminator. And again, don't be confused by these terms. They're just parts that are that is genetic code. And here, so these parts can actually be cut out and then glued together. And the kind of like putting together pieces of Lego or whatever, connects, I guess, if you played with that as a kid. And we need something to cut our DNA, and we also need something to glue our DNA back together. What we use to cut DNA is called, we call restriction enzymes. And what they do is they cut at specific codes. So remember the A's, T's, C's, and G's. These restriction enzymes will actually find a specific sequence, order of A's, T's, C's, and G's, and cut at that specific spot. And then under the right conditions, these will actually come together, and then what you can use is cellular glue, which is ligase, and that'll actually, because when you cut the DNA, it cuts it at that phosphate connection to that sugar, if you kind of recall from like 10 minutes ago. And what the ligase does, it reforms that bond so that your DNA doesn't come apart again, because it comes together kind of like, because the chemical bases will actually stick together, the A's, T's, C's, and G's, like specifically like Jen was talking about earlier. But the backbone, the phosphate to the sugar, isn't connected, so that's what the ligase is for. That make sense? Okay. Okay. So here, this is the general scheme of how they actually string together parts they want to in whatever order they want to. So just think of this as part 1, this is part 2, you want to put this part in front of this part, so you get this piece, okay? I'll go through this in a little bit more detail in the next few slides. Essentially, you have 2 pieces of DNA, or two plasmids, each with the part that you want to put together, and through a process of using restriction enzymes and ligases, you can actually make a plasmid with the 2 parts you want beside each other in that plasmid. So we'll go on to the biobrick assembly. So essentially, here we have 2 parts. Let's call one part 1, one part 2. And if you just imagine, this just kind of extends all the way back around. You have the plasmid, so you have 2 plasmids here. And they have antibiotic resistance, again, for selection. So we have this part, we want to put that in front of this part, okay? And these right here, they're just names of restriction enzymes, and their name is actually just based on what organism they were discovered in, and what number. And by number I mean, because there are more than one restriction enzyme that would come from one organism. So like if you look at EcoR1, that's from E. coli, ECO. R is actually the strain of E. coli, and then 1 is the first one they found. But that's not super important. So here, this might be kind of scary, but these are the chemical codes of DNA, and each of these cuts specific codes that are different from one another. Okay? And you don't have to memorize them or anything, but I want you to notice something, and that's with these 2. Okay? So we have SPI1, which is at the end of part 1, and we have XBO1, which is at the front of part 2. Okay? So now, with restriction enzymes, they cut at what are called palindromes. Does anybody know what a palindrome is? It's not a scientific word, so.
Reads the same
Yes.
form.
Sorry?
It reads the same form backwards.
Yes, exactly. Can anybody give me an example of one? Yeah. Okay. So if you read this way, it goes HCTAGT, same that way. Remember the 3', 5' directionality anti-parallel? So now, each restriction enzyme cuts at a specific palindrome. Now, the important thing about these 2 is, so the arrows show where it cuts, so it'll actually cut like this, leaving this A, so it'll cut kind of like that. And the thing I want to point out is the middle part, the CTAG on this side, actually is the same as this side. Now, they don't cut the same site because the 2 nucleotides on, like the one on the left and the one on the right, they're actually different for the 2 restriction enzymes. Does that make sense? Yeah. Okay. That is very important for later, so make note of that. And so I'm just gonna go to the next slide, and essentially all I did was I moved part 1 over this way, and moved part 2 up to here. So just kind of follow that. And so now we're gonna cut our DNA. Okay? So here we have our XBA from earlier, and this was SPI1. I just kept the sequences down here just so you can follow it. So colors, it'll make it a little bit easier to follow. So this is a mirror of the top, okay? So essentially what will happen is our restriction enzymes, when we add it, will cut our DNA, just like this, okay? So, like I said, it would cut like that, and again, because these nucleotides, C's match up with G's and T's match up with A's, we can actually bring part 1 and part 2 together. And they'll do this by themselves just because C's like G's and T's like A's, essentially. So if under the right conditions, they'll actually come together. But now we still have that problem with the backbone, which I don't have here, but like the sugar phosphate, the bond between, so that there would be a bond between these two bases, right? So it'd be the bond here. It's still broken, and that's why we need ligase. It's gonna make that bond again, and the DNA will become whole. Okay, and so that's the genetic loop, and we call that DNA ligase. So essentially, now we have a whole piece of DNA, and what I want to point out is if you compare this to the 2 sequences we started out with, we had this sequence, which was cut by one restriction enzyme, XBEL1, and this one, which was cut by the other one, SPE1. And if you look at them, I think, no, I didn't. Okay. So this new sequence that we have, it came together because the middle 4 match up, but this side of this sequence is the same as the XBEL one, and this side is the same as this is XBEL. So this, just note that this side is the same as this side, and this side is the same as this side. And that makes sense because the 2 parts came together. And the reason why I wanted to point this out is because with this, you can't cut this with either one of the restriction enzymes we used earlier. And what we call this the star sequence. So you essentially added part 1 and part 2 in the order you wanted it to, okay? But now if you wanted to, you can add part 3 to either end of the piece of your BioBrick that you want. And when you add either 2 of the restriction enzymes again to cut it and bring them together, then it won't cut here again. And because of this, we can actually keep adding and build a chain of genetic code in the order we want. Does that make sense? So after we have let's assume this is part 1 and part 2 together, and it's on a plasmid. Pretend there's some type of antibiotic resistance, so we can select for it. Okay, so we have our piece of our plasmid with our BioBrick in it. Now, Jenna mentioned earlier that there's a process called transformation, where you actually put the DNA into the cell by having special conditions like heating up the cells to a certain temperature, and that'll essentially cause its membrane to kind of open up and it'll grab stuff from the environment, and then the DNA can actually go into the cell. And then from here, you can actually, because you put all this work into making this DNA, you deserve a break. And what you can do is you can have the cells grow for you. You just put them in some media, put them in an incubator at 37 degrees, leave it overnight, you come back, you have a whole bunch of cells with the DNA that you made earlier. And the reason why they'll have this is because you have that antibiotic resistance that will select for that, for the ones that actually have it, if that makes sense. So going back to ampicillin, if you had ampicillin in your media and you put these cells in your media, all the ones that have this piece of DNA in them will be able to grow because the ampicillin can't take them out. Okay, and then there's also another lab technique that is called a mini prep that you guys will be using. And what a mini prep is it's essentially getting the DNA from your cells. So you grew your bacteria overnight, and they all have the DNA that you popped in and then duplicated. So you essentially, there's a little bit more detail than this, but I'm just going to touch on it briefly. So you essentially first open the bacteria cells so that you can get access to the DNA, and then you run everything through a filter that specially binds the DNA. And because of DNA's properties, you can actually wash all the other junk through it without taking out the DNA, because the DNA will be stuck in the filter. Okay? And then when you want, you can add this solution, which is actually usually water, and you can take the DNA out of the filter. So you have your bacteria, you burst them open, you run it through a filter, the DNA sticks in the filter, you wash it with ethanol actually, and then you add water and it'll take it out of the filter, and then you have a tube of DNA in water, and that's it. Okay? So you have purified your DNA. And this is a really quick process. It actually takes about like 20 minutes. Okay, so after you have your tube of DNA, you want to be able to tell it is your DNA. There's two ways you can do that. You can spend lots of money, send it away, and they'll actually, they have a process where they can actually read every letter of what the sample you send them, and then they'll send it back to you. Or if you know, because you know the size of your parts, and by size I mean number of nucleotides that you put together. So if you think about it, you have two parts, and when you put them together, the end product will be bigger because you have 1 plus 1 equals 2, right? And so when you run something on the gel, you're actually separating it by size. So I'm just going to explain an agarose gel to you. So what happens is, if you remember, the DNA is plasmid DNA, so that means it's circular, so it goes all the way around. And that kind of becomes a problem when you want to separate stuff by size, because it can get twisted up, and it can get twisted up with other pieces of DNA as well. So you can use any of those 4 restriction enzymes that I pointed out earlier, and it'll cut it. So you cut the circle, it'll turn into a line, essentially. You have circular DNA, cut it once, it'll be linear DNA. And then, because all your DNA is linear, you can now run it on a gel, and it'll separate it by size. And the concept behind a agarose gel is essentially DNA is negative. So if you put DNA, if you run a current through, we all know negatives and positives attract, right? So if you make this end of the gel positive by an electric current, and you put your DNA up here, your DNA will go to the positive end of the gel because it is negative. And so how the gel works and how it separates things by size is essentially the gel itself is a matrix, and what I mean by that is, if you imagine you're at school and you have to go to class. So you get out of the classroom, you gotta go to your classroom. So if you were to go, if there was no one in your school and you walked out of your classroom, it'd be pretty easy to get from one end of the hall to the other end of the hall. But if it was class change time, then there's a whole bunch of other people there. So they're kind of in your way, and then it takes longer. So if you were a smaller person, you could get through the crowd of people quicker than if you were a bigger person. So if you look at this gel, the pieces of DNA that actually appear higher are actually bigger than the pieces of DNA that are lower. Does that make sense? Yes? Okay. So based on that, because with iGEM we work on the genetic level, so the DNA level, and then that later will allow us to measure activity of our proteins and stuff.